Good morning, everyone, and welcome back to the virtual international conference on global research trends in biotechnology, hosted by the Department of Biotechnology from St. Joseph's College of Engineering. It is my privilege to welcome our speaker for this evening, Dr. Venkatesh Endalur Gopinarayanan. He is a young yeast biologist with over 7 years of experience in genetic, protein, and metabolic engineering. He completed his BTech in St. Joseph's College of Engineering and his MTech from Anna University, majoring in biotechnology. During this time, he was an undergraduate research intern at the National Institute of Epidemiology at ICMR in Chennai. He then went on to obtain his doctorate in biotechnology engineering from Tufts University in Medford with an astounding GPA of 3.97 on a scale of 4. While there, he worked on constructing a yeast platform for catabolism and any non-native carbon source and engineered synthetic regulons in Saccharomyces cerevisiae for non-native substrate catabolism. He has also trained and mentored 3 undergraduate students at Tufts University. He is the recipient of various awards and fellowships, including GATE, MHRD Government of India Graduate Scholarship in 2011, and the Shirley and Stanley Charm Scholarship in Food and Biotechnology for both the summers of 2013 and 2014. He also received the Graduate Opening Access funding in February of 2018. He is a semi-finalist in the Dell Social Innovation Competition conducted in March of 2011. He has published papers in leading journals such as Nature's Communications and Biotechnology and Bioengineering. Currently, Dr. Venkatesh is a scientist at LifeMind Therapeutics Incorporated and has been instrumental in engineering exotic fungi at this company. He has developed synthetic orthogonal expression systems for production of small molecules, designed and built multiple synthetic biology tools for engineering model and non-model filamentous fungi, and has also worked with a collaborative team of scientists and researchers on multiple projects. Sir, it is an immense pleasure to have you with us today. I request you to take over the session.
So as I was saying, it's definitely, I'm definitely glad to be back and especially giving a talk at St. Joseph's College of Engineering. And without further ado, I would start my presentation. All right. So this is one of the major chunk of work I did during my PhD for like 4 or 5 years. We were working on this project. I personally felt it's a good project to speak to and mostly undergrad audience, since I thought this as a synthetic biology field combines a lot of topics and courses that as undergrads and even postgrads, MTech students study. I felt this combines a lot of biochemistry, genetic engineering, reaction engineering, biochemical engineering, basically like entirely diverse fields, and brings them together and makes it into like a nice research. And also I thought this is a good way to show that, whatever we read during our undergrad and, masters are not just because they are theory, right? They are theories that can be used to solve world problems, actual practical world problems. So that's basically the reason why I'm here presenting this topic. So I am a scientist at LifeMind Therapeutics, which is a biotechnology drug discovery company in Cambridge, Massachusetts, USA. Since a lot of the work that we do there are kind of confidential, I can't reveal those. But yes, we do engineer a lot of different fungi, and it's been like a very exciting journey for me over there. But what I can talk about is my PhD research, which also has the same flavor as what I currently do right now. Before I talk about my research, I'm sorry, I'm trying to move my slides and, okay, sorry, yeah, no, it's fine. I would like to actually acknowledge a lot of people who has helped me with my research, and especially the lab that I worked in during my PhD. I would also like to point out the advisor is Dr. Nikhil Nair. He has been a huge help for me, and without all the support of people who were there working in the lab, this wouldn't have been possible. And finally, I was just speaking to one of my advisors one of the time, and it seems that they're really sad that they don't get a lot of applications from India. Tufts University is in Boston, and since it's a small research university, it doesn't like to publicize itself, and the rate of acceptance is also like very low. But still, it would like to see a lot of applications coming from India, especially from Anna University. This has been personally communicated to me by the chair of the Department of Biochemical and Biological Engineering, also by my advisor. So this is just a quick snapshot of how the university looks like, how the labs look like. So if anyone is thinking of applying for higher education, please go ahead and give it a try. They're a good university. I know a lot of people haven't even heard the name. So I thought I'll just shamelessly publicize it for like a minute and then go on to the research part. All right, going on to the research part. I know this involves a lot of words that people are not, sure of or haven't come across. But basically, what all these mumbo jumbo translates to is: can we make yeast eat whatever that we wanted to eat? Yeast are generally yeast, not just any yeast, but the baker's yeast or Saccharomyces cerevisiae are not non-picky eaters. They are kind of fastidious in what they eat, and I would like to go ahead and translate and show why they are like they are. So the reason why this project, I just like to give a little bit of motivation before I go forward, right? So the main motivation is doing better metabolic engineering, and as a field of metabolic engineering, it has been there for almost like 3 to 4 decades. And the idea behind that is basically you take an organism and try to engineering it in different ways, and you make chemicals or drugs, fuels, etc., so that it can be used in an industrial scale. So the whole idea of metabolic engineering can be broken down into basically you giving some kind of a carbon source and other nutrients for microbial cell factories, and that gets converted to some kind of a product later. So that's all metabolic engineering really is. And the products basically can be, anything from, industrial bulk chemicals, fuels, drugs like antimicrobials that I just talked about, or even like fine chemicals that are like super expensive. And it also depends on what the feed you give it. And it can vary anywhere from, lignocellulosic biomass, which is basically like, the most abundant biomaterial that's there on Earth, which in turn is composed of cellulose, hemicellulose, which in turn has, other monomer sugars in there. It can also be, made of other sugars like lactose, single carbon compounds like alcohols or aldehydes, like methanol, methane, except formaldehyde, like that. You can also use carbon dioxide, which would, result in a absorption or, retake of all the carbon dioxide that's out there in the atmosphere. And the products, right, that can be split into different sections. You have either your low profit margin products, like, industrial bulk chemicals, biofuels, etc. And you have your high profit margin products like, antimicrobials or fine chemicals. If you have your high profit margin products, you basically don't have to worry about what kind of sugar you put in. You don't have to worry about the cost of the entire fermentation and the process itself, because you'd be having a high enough profit margin that to cover, even expensive sugars. But in the case of the low profit margin ones, then you need to really care what kind of sugar you put in. So then the question kind of reverses on itself, basically. So the question becomes, what kind of sugar that is cheap enough can be used, for metabolic engineering, so that the entire process becomes viable and feasible. In that setting, the cheapest one that you can think of is lignocellulose, which is, as I said, basically wood particles that has been treated and has like, a lot of other sugars in it. And since it's the most abundant biomaterial on Earth, it's like easy to metabolize, or sorry, easy to find, but not easy to metabolize. Very hard to metabolize, actually. Uh, yeah, so as I was saying, lignocellulose is, ideally the best nutrients that we want to use. And as it has multiple sugars, whenever you want to, metabolize lignocellulose, you want to make sure all the sugars that are there in the lignocellulose mixture is also, completely metabolized, because you don't want to have like 30 to 40% of your, lignocellulosic mixture having sugars that are not metabolized at the end of your fermentation, which is going to directly affect your, productivity downstream. And in our test case, we wanted to see what people have been doing generally. Since a lot of these sugars are not metabolized normally by most of the microbes, there has been a separate field called catabolic engineering, where people try to engineer these organisms so that they utilize these low-cost sugars really well. So that is the field of catabolic engineering in essence. So how do we do that? Is that you take a sugar that is not normally metabolized, which I'm going to call it as a non-native sugar, because that particular organism cannot normally natively metabolize that sugar, and you want to make it assimilated into its native pathways for any of the industrial microbes that we have. And how do you do that? Is basically you start expressing enzymes that are not native to these organisms, so that you convert this non-native sugar into some kind of a native metabolite by going over any kind of non-native intermediate that is there. So once you get into a native metabolite that's there, then it kind of feeds into any of the central carbon metabolism chain that's there, like, glycolysis, pentose phosphate pathway, PCA cycle, etc. So once it feeds into that, then, the native metabolism takes over, and then you start you can get your product as well as, biomass for growth. But usually whenever this process happens, it's usually not that smooth. There's a lot of issues in there. We basically see a lot of thermodynamic limitations that's there. We see limitations with enzyme kinetics. We see that the Vmax or Km of the enzyme for that particular non-native sugar or the non-native intermediate is not great, especially because it's in a heterologous, system where the pH and temperature for that particular enzyme is completely different from what is there for the native organism. So then you need to start engineering these particular steps to basically start bottlenecking it, or removing the rate limiting reactions that are there. But as we all know, in metabolic engineering, there's not just like one rate limiting reaction. As you remove one, the other one pops up. So, you start to do it iteratively. And in an ideal case scenario, you want the growth rate of your organism in this non-native sugar to be as high as how it would be in like glucose or some kind of a sugar where it grows really well. So people have been, as I said, doing this field for the past 30, 40 years. And instead of doing this iterative, process where, you start engineering each and every enzyme that's there for ideal performance and increase the growth rate, what people tend to typically do is start accumulating beneficial mutations in these organisms by selecting it for growth in these non-native sugars. And this technique is usually referred to as adaptive lab evolution or adaptive evolution. And that's just like a crude way of saying, I don't care what's going to happen inside the cell as long as it grows better in my particular native sugar that I want. So what I'm going to do is just start serially growing them again and again for like a few weeks or few months until the growth rate of that particular organism in this particular, non-native sugar is like really high. So when I started doing my PhD, I was really interested in this field of metabolic engineering and catabolic engineering, especially because this particular field of catabolic engineering has been like really recalcitrant and not easy to work with. As you can see, you need to do like extensive engineering in this to even make sure one particular non-native sugar is being consumed. And yes, there are examples of successful engineering in this field, but it involves not you just express a few enzymes and that's it. Voila, you're going to get your final product. That has never been the case. It always has involved doing extensive rounds of mutations, serial adaptive lab evolution, along with expressing these pathways. So in this case, I thought, why is it so hard? Why is this problem so hard? It should be like easy for us to, solve this. So we thought, okay, let's, try doing something different for this project to see if we can, solve it in a better way. And also another thing that we found was quite shocking is in the past 20 years, there has been like no new tools or strategies, that has been newly available in this field. So we wanted to address all these problems that are there. And as a test case, we wanted to solve an issue that's the most, studied problem in this field, which is basically xylose metabolism in Saccharomyces cerevisiae. So we thought, let's take this as a test case and work with it. And Saccharomyces cerevisiae or yeast, is, a commonly used industrial workhorse. And xylose is the second most abundant sugar in lignocellulose that I just talked about. So naturally, people want to make sure that xylose is utilized by yeast. And because of that, there's been almost like 3 decades of research in this field. But even after 3 decades of research, the fastest strain that, has been designed rationally without using adaptive lab evolution only grows at a rate of 50% of the growth rate as that of, when you grow it in glucose. I think in glucose, the growth rate is somewhere around like 0.3 to 0.35 per hour. And in xylose, the maximum that was engineered was like 0.15 or 0.13. So, this is not an ideal case scenario, and we definitely would like to, increase it. So then how do people usually engineer it, right? So people can engineer it researchers have been engineering using 2 different pathways. So I have those 2 different pathways here on either side. And the one on the left is basically referred to as an oxidoreductive pathway, as you can see here. Xylose is being reduced and then oxidized. So basically it goes from a sugar, aldehyde sugar, xylose, to an alcoholic sugar, xylitol, and then you convert it back to a ketone, xylulose. Or you can just use an isomerase and directly do an entire conversion from xylose to xylulose. Finally, you add in a phosphate molecule to it. So you have your xylulose 5-phosphate. It's charged. It's ready to go into the pentose phosphate pathway. So once it enters pentose phosphate pathway, it directly feeds into central carbon metabolism and then it goes ahead. So these has been like the 2 ways to engineer it. And we actually thought we would go over with this right side pathway of the isomerase pathway using a xylose isomerase and xylulose kinase to do our engineering. But before we went ahead and started engineering, we wanted to see, okay, this is how we as humans have been engineering these, right? And also when we try to engineer this, we want to make sure these particular enzymes that we are, genetically inserting into these organisms' genome is constitutively on. So they don't have any kind of regulation in there. They're always turned on. So we thought, okay, how does nature metabolize this sugar? And because yeast in itself metabolizes several sugars on its own, and it does it very well. It is known for, converting sugars into, products like acids and alcohols quite well. So we thought, okay, let's study how nature does it, especially how does yeast do it, and can we take lessons from that and apply it towards our engineering step. So when you look at natural sugar assimilation, especially in higher eukaryotes and eukaryotes in general, it's not a process of where you have these enzymes that are being turned on all the time. Even let's say in lower order organisms like, prokaryotes like E. coli and all, you still have like operons and things like that. I'm pretty sure people have studied about like, lac operon, where in the presence of lactose, you have your genes turned on. So similar to an operon, but higher and larger order in scale is something called a regulon. A regulon is basically where you have dynamic regulations where hundreds of genes are being regulated by a particular transcription factor in response to some kind of sugar or any kind of signal that's there in the environment. So let's say you have some kind of sugar like, galactose, glucose, xylose, etc. This particular sugar is being sensed by a native sensor that's there in these organisms. And that in turn would turn on the transcription factor. And this transcription factor goes and binds to all the genes upstream regions of these genes that are needed for growth, and then would turn on all the necessary pathways that are needed for, better growth, for making biomass, for making products, etc. And one interesting thing that we found is that, especially in yeast, these regulons are, highly object-oriented. For example, let's say there's a glucose regulon. So the main purpose of glucose regulon is for completely fast glucose depletion. So you won't have that much biomass, but you're going to have a lot of alcohol in there. Versus on the other hand, if you take galactose, it has been highly tuned for actually, better growth rather than forming any kind of a product. So you can see here that these are actually not, random genes that are being upregulated all the time. They are, specific ones. So we wanted to take a look at it in detail and have a snapshot here of the galactose regulon in yeast. So it basically follows the same principle as I talked about before. Whenever you have galactose in the environment, it is being sensed by two kinds of sensors. There's a primary sensor and there's a secondary sensor. Both of these sensors basically bind to galactose, and that results in a conformational shift in this particular protein. That in turn, activates this transcription factor called Gal4P. And Gal4P is like the primary transcription factor that goes and activates all the genes that are required for growth on galactose. So we went ahead and looked at what kind of genes are these that are needed for growth on galactose. Can we take, a deeper look into it? So we found that it can be divided into two kinds of genes. One is something what I call as the initial metabolic genes, and something what we call as downstream genes. So the initial metabolic genes are basically the Lelluar pathway, it's called, where basically glucose is converted, galactose is converted to a central carbon metabolism intermediate, right? So galactose goes from galactose to galactose 1-phosphate to galactose UDP, and then finally it goes from glucose 6-phosphate in the end. So that is basically the initial metabolic genes. And then there is something called the downstream genes. We know this because we know that when you activate Gal4P, a lot of genes gets upregulated. But we don't know the role of all of these genes that are there. So then the question becomes, what do the genes do? Why are the genes needed to even, get upregulated for growth? So we went digging a little further, and we did find that hundreds of genes are upregulated. And when we looked at the functions of these genes, we found that a lot of genes are required for cell growth, mitochondrial function, protein synthesis, basically things that are actually needed for growth. They are not, random genes that are being upregulated. And when you take a look at these particular genes that are, up or down regulated, it seems that it doesn't matter what kind of sugar you grow it in. These genes are, required for growth in all kinds of sugars. So we thought, okay, now let's compare what we have with the galactose sensing system in yeast versus what we as humans have been engineering for xylose. So the first thing that's, quite obvious is that we don't have any kind of a sensing system with xylose, versus with galactose, you have this like highly precise nutrient sensing system that's there. As I said, since we don't have any kind of a sensing system, interestingly, whenever people have been doing transcriptomics in it, especially for growth in xylose, they do find that, cells actually think that they are starving. even though it has a lot of sugar in the medium, it cannot sense that there is a particular sugar because that xylose is not in its natural habitat. It's not found. So it starts thinking it's in a starvation mode and starts, upregulating all the genes that are required for, whenever there is, the absence of sugar. So it essentially, even though it's consuming a lot of sugar for itself, it goes into a starvation mode, which, as you can, guess, is not ideal at all. The other thing is that galactose growth is, Regulon control. It's, highly specific for a particular objective, which is for growth in this case for galactose. In xylose, we don't have any of that, fine tuning of the pathway. We are just going to overexpress a couple of genes, and then we don't have any regulation of it that at least we know of. So you can think of it as some kind of, the metabolic pathway as some kind of tubes connected with valves, and you have like an overhead tank, which is controlled by this master regulator or regulon. So when you have this galactose regulon that is turned on, it kind of goes and starts turning on other valves or other transcription factors. So you end up producing whatever. In this case, it's just biomass and then one particular product. It doesn't produce random things. However, let's say you have your xylose metabolism that is not engineered. Then in this case, there are no specific regulons, and we don't know how these transcription factors are going to behave. So what you end up is having is that there's this bleeding of flux through these pathways to different products and biomass. You don't get any specific one; you just get like random ones. And in engineering, you definitely don't want that. You want to push your flux or push your pathway towards one particular product or biomass that you want. So we thought, that kind of makes sense why we've been having so much trouble with the xylose pathway for this long. And as I said, the third one is basically reiterating the fact that, there's regulon-based dynamic control in galactose. In xylose, we don't have that. Okay. So that basically goes directly over into the question of, so what does this regulon do and how does it, affect growth? So we wanted to, test that out and actually show that a regulon is actually very important for growth by using this galactose as a test case. So we have this wild type strain of yeast that has this intact regulon in it. And then we have this galcons that we call it as a galcons, where we knocked out this transcription factor called gal4p. So once it's knocked out, we took these Lelluar pathway genes, the genes that are needed to convert galactose to glucose 6-phosphate, which is a native intermediate, right? And started expressing them constitutively. This is basically our own version of what would happen if galactose cannot be natively used by these organisms, and we are going to engineer galactose metabolism in yeast. So then at that point we wouldn't have any transcription factor specific for galactose, so we knocked that out of the system, and then we started expressing these initial pathway genes or initial metabolic genes called Lelluar genes. And we call this as the galcon strain because it's constitutively expressed galactose metabolism. And we don't know what happens to these downstream genes in this. And we thought, okay, now let's compare growth between these two strains. And as you can see here, the wild type strain, the gal reg that's there, has a much higher growth rate than the galcon strain. The wild type strain has a growth rate that, what we have been what we have seen in other literature too. It's 0.3 per hour, completely normal, but you can see a huge lag that happens in the galcon strain, which kind of proves our hypothesis in the first place that you need these downstream genes and this gal4 to be there. But then we had a question, is it a difference in expression that's there in these Lelluar genes that's causing these, or are there like, things that are, messed up because we started engineering with the system? So we thought, let's put back the gal4p in, but we'll still not have the LILUR genes under the expression of these GAL4P. We'll still constitutively express these LILUR genes and have GAL4P expressed so that it can activate all the downstream genes. So this is kind of an in-between version of what a wild type strain is and what a GAL-cons is. We call it GAL-cons, GAL4. I specifically don't know why we named it like that, but thinking back, I think it's a weird name for this particular strain, but whatever. So with that particular strain, you can see that it actually recovered a lot of the growth rate defects that we found in the GAL-con strain, just because we have GAL4P being expressed, which in turn starts expressing, which in turn starts activating all the downstream genes. So this kind of confirmed our hypothesis that showed that the Regulon-controlled downstream genes do play a huge role in growth. And the second thing that we actually assumed, and we also confirmed it with like transcriptomics, was that there are a lot of downstream genes that are not specific for galactose metabolism that is being upregulated whenever you have this GAL4, the transcription factor, being upregulated or being turned on. So using this knowledge, now that we know that, okay, these Regulons do play a huge role, can we use this knowledge and apply it for, xylose metabolism? That was our second part of this. We thought, okay, let's now build a synthetic xylose Regulon. Because so far people have been, constitutively upregulating genes for xylose metabolism. And there hasn't been any study on, or very little, no study or done on, xylose regulation. But in this case, we thought, let's build xylose regulation first and then worry about metabolism second. So then that becomes a question of, how do you build these xylose regulons? In an ideal case scenario, it would be a bottom-up engineering where, you would have a sensor for the xylose, which in turn would activate a regulator, and all the genes for growth on xylose would be activated. But as you can guess, again, this would involve extensive engineering where you need to find a sensor, couple it to a regulator, and then start engineering all the genes for growth on xylose. So we quickly decided not to do this and thought, let's do a top-down approach where, since we already know this galactose regulon works really well, let's take this galactose regulon and repurpose it for a xylose regulon. So in that case, what we have to do is we have to take the first part, which is the sensor that's there, which is called the GAL3 or the GAL1, and engineer it so that it can sense xylose instead of galactose. So that will be the first engineering step that we need to do. Second is we need to mimic this signal transduction that happens with galactose in the case of xylose metabolism or regulation. And finally, we need to place genes that are needed for growth on xylose under this control of this GAL4P transcription factor that's there. So we thought, okay, let's start going over each of these and start engineering them. So first is to make this particular sensor GAL3P respond to xylose. So for that, we have to take this protein and start mutating them so that it can bind to xylose. So how we did that is basically we came up with a screening and selection system where we thought, you can think of this entire system as some kind of an AND gate where you have your active GAL3 that's there, and then you have your galactose or xylose. And only when both of these are there, they would, result in either antibiotic resistance, which would be offered by this CanMX gene, where we would be giving it some kind of an antibiotic. And then we would have green fluorescence being upregulated, which you'd be basically seeing it as fluorescence, which can be measured and quantified. So we built this, screening and selection system that's there. And since we have antibiotics, it can be easily selected for in a plate that has antibiotic, which is CanMX or G418. And then we can measure what the fluorescence is to get a quantitative output. So what would happen is, as a control, we can, give galactose, see what is the antibiotic selection, see what is the fluorescent readout, do whatever mutation we want with gal3 protein, and then give it xylose and see is there antibiotic selection, is there fluorescence readout. So that way we can keep track of, is it working well in galactose, and can we start seeing, because of these mutations, is xylose binding to gal3, and is that causing some kind of an antibiotic selection or a fluorescence readout? So that was basically our entire selection screening system. I'm not going to go over all of them, but we kind of screened like 500,000 mutants. And finally, we did get what we wanted. We did 4 rounds of different kinds of mutations in it. We did something called an interaction loop mutagenesis. I can go over that later after the talk, because you might be running over time. And then we did site saturation mutagenesis. Finally, we thought, let's start mutating them randomly. And then we took all those different kinds of mutations we had and started shuffling them using a technique called synthetic shuffling. And finally, we ended up with this final mutant, which we call as gal3 syn 4.1. As you can see here, the wild type does not bind to xylose well. It has a really low preference or really low fold change at 2% xylose, which is nothing but fluorescence of that is observed with and without xylose. And the final mutant has, 15-fold higher this fold change than what we saw in the wild type gal3. And that's basically the raw fluorescence value that I'm showing over here. As you can see, the gal3 syn 4.1 worked as we wanted. So we were happy with that. So we thought, okay, with 4 rounds of directed evolution, right now we have a good higher interaction strength of, this particular gal3 protein with xylose. Let's go ahead and start the next step. It's basically we want to mimic this signal transduction that happens with galactose and xylose. So I know I've shown this figure a couple of times, but what essentially happens here is that there are different feedback loops, like interconnecting feedback loops in this regulation, right? As you can see, there are like negative feedback loop by gal80 towards gal4. And then there are 2 positive feedback loops, one created by this gal3 protein and one created by this gal1 protein. So the difference between gal1 and gal3 is that gal3 is expressed at a higher basal level, even when galactose is not present, versus this low gal1 is like it's actually under strict tighter control than gal3. And people have done a lot of simulations on this, and what they came to find out is that the reason why we have 2 different kind of sensors is so that this kind of a dual feedback loop results in increased sensitivity first, and then it also lowers noise. So we wanted to engineer a system that basically has the same function. You wanted to increase sensitivity and lowers transcriptional noise in the system. So to do that, then we have to engineer gal1 next. The issue with gal1 is that gal1 is also acts as it's actually an enzyme that moonlights as a transcription factor or transcriptional binding sensor. Then in that case, trying to engineer gal1 is not a good idea, because it is also an enzyme which is going to bind to galactose and converts it to galactose 1-phosphate. The issue with this particular enzyme is that galactose 1-phosphate, the metabolite itself, is a toxic metabolite when it accumulates to a higher amount. So you would expect that if it in case starts cross-reacting with xylose, you might end up having xylose 1-phosphate there, which would be toxic for the cell. So we didn't want to play around with that. We didn't want to even engineer this galactose 1-phosphate. But we kind of assumed that, since we know what the role of galactose 1-phosphate is, and it seems the main reason why it works the way it works is because it has this low basal expression when galactose is not there, or it has a tight control. And then when you provide galactose, it has a higher expression. And that is what is needed for this low noise system that's there when galactose is present. So our idea is that let's keep galactose, let's keep gal1p as it is, not touch it. but we'll take this gal3 protein and express it in the same way. So instead of having one gal3 protein, we'd have two different gal3 proteins. This is basically what I talked about. Sorry, I forgot to skip a slide. So instead, we thought that we would have two different versions of this xylose regulon. One is something called a single feedback xylose regulon, where we would completely remove this galactose 1-phosphate that's there. And second is we'd have a dual feedback of this xylose regulon, where we would have the same gal3p mutant being expressed the same way as how gal3 is expressed and also how gal1 is expressed. So emulating that kind of the feedback regulation of a galactose regulon would give us increased sensitivity and also minimize noise. So we tried doing that. So we came up with this dual feedback system, and then we looked for sensitivity. So basically, I'm giving so it's the same selection system or screening system that I talked about before, where increasing concentrations of xylose is given and fluorescence is being checked. And you can see the dual feedback loop has higher fluorescence than single feedback for the same concentration of xylose that's used. We were happy with that. Then we wanted to see does it also stabilize the expression. So we wanted to look at what is the coefficient of variation when this thing is when a different concentration of xylose for the same fluorescence. And again, you can see here that there's a consistent decrease in coefficient of variation in a dual feedback loop than a single feedback loop. What it basically says that if you have a dual feedback loop, it's going to have a more stable expression than when you have a single feedback loop. So again, it seems clear that as long as you have, especially in like low concentrations of xylose, there's like a very stable expression. So we're happy with that too, and that leaves us with the final step. We want to actually engineer metabolism now. So we thought let's create a xylose regulon strain where you would have xylose activated, which would be sensed by this gal3 4.1. which would in turn activate gal4p, and you would have which gal4p would then go and bind to genes that are responsible for xylose metabolism and turn it on, which the strain we call it as a Xyltrex strain. But then we wanted to see, okay, so now we have this regulon strain that's going to, hopefully performs better, but then we need something to compare it with. So we thought, let's build this Xylcon strain, which is, which is basically a strain that doesn't have downstream genes, but has all the xylose metabolic genes being constitutively overexpressed. And we thought, let's compare it and see which one performs better. And there's no surprise here, as we expected, as we saw in the case of galactose, the Xylrex strain or the xylose regulon strain performed much better than the Xylcon strain that we saw. So we were all like, really happy. We were like, yes, finally we got what we wanted. But as a scientist, as a student of science, you keep wondering, yes, you did all this, but what is it that is actually making it grow better? We want to, dig into it further. So that's the next section of the talk. But just to go over what the summary of this section is, that we kind of confirmed without doubt that Regulon is needed for growth. So we thought, let's engineer a synthetic Regulon in this case. And to do that, we had to do some protein engineering, metabolic engineering, some regulatory pathway engineering. Finally, we obtained this Xyltrex strain that seems to be growing much better than Xylcon strain. And coincidentally, we found that that is the fastest growing rationally engineered xylose strain that we had. We were happy with that. Then we wanted to know, as I said, why do these strains grow faster? So we thought, let's take all the Regulon strains that we have and all the constitutive strains that we have and, pit them against each other and do like an RNA-seq analysis or transcriptomics analysis. So here I'm just having a table of, growth of different, of all the strains that we have, how they are grown, what is the regulation involved in it, and just a comparison of their growth rates. So we took all these strains, performed RNA-seq on it. Just to go over what RNA-seq is RNA-seq is basically looking at the RNA as a substitute for looking at protein. And the idea is that the different levels of RNA would give an estimate of how much the genes are being up or down regulated. So this is basically just a crude heat map of all the genes that we had, sequenced and compared. There were like around 50 to 100 genes that were analyzed. We thought, we'll take like a little bit of systematic approach. Here, we thought we would, find out differentially expressed genes. We would see what are the genes that are upregulated in these red strains and what are the genes that are upregulated in these corn strains, and then group them based on function. And then finally ask the question, okay, now that we have all these data with us, does it actually mean anything? So we found that around 450 genes were upregulated in the Regulon strains, and around 500 genes were upregulated in the, those corn strains, which is a way of saying 500 genes were downregulated in the Regulon strains. So then we took all these genes and started grouping them based on functionality. And we found that all these genes that are upregulated are actually growth-related genes. They are needed for growth. No surprise there, I guess. What we also found that we thought is quite interesting is that a lot of these genes that are response for stimulus-based genes are actually stress response-related genes. And some of them are also, starvation response genes, which I think I initially mentioned much earlier that, that is a phenomena that seems to happen whenever there's no regulation involved in growth. So the same thing is here, where these corn strains are thinking that they're actually starving because you don't have any sensing system for these sugars. So once we did all this, it seemed it is clear why we see an upregulation of growth or increased growth in Regulon strains. But then we wanted to know, are we seeing these increase in growth regulated genes because they are, growing better, or it's growing better and that is what is causing growth related genes? It's basically a question of, is the growth related genes increase in upregulation we're seeing? Is it a cause or an actual effect of increased growth? To do that, there are two things to do. One, you can look at metabolomics, which at that time our lab was not equipped to do. But we can also look at growth related transcription factors and see whether they are on or not. So that we thought that is a potentially easier way to track it down. So we started looking at all the transcription factors that are there. And I'm just skipping a little forward here. And so there are a total of 184 regulated transcription factors in yeast, out of which we found that almost 39 were upregulated in either the reg or corn strains. So we took these 39 transcription factors and analyzing what they do. And the same pattern emerged, basically where some of these transcription factors upregulated in the corn strains are responsible for stress and nutrient starvation. And then the ones that are upregulated in the red strains are responsible for growth. Just to summarize whatever I spoke over here, transcriptomics basically gave us more conclusive evidence and showed that whatever the observed phenotype is there is because of Regulons, and that when we do this differential gene expression and transcription factor analysis, it seems clear that reg strains have a lot of growth-related genes that are being upregulated. Specifically, we found that a lot of cellular and mitochondrial biogenesis pathways to be upregulated, and that is possibly what is being translated as better growth in these strains. How are we doing on time? Okay, we have like 12 minutes. I'm going to quickly go over something that we did as an afterthought, which is basically trying to expand this Regulon engineering that we developed. So the idea is that this arabinose metabolism, right? It's an other sugar, second most abundant pentoses that are there in lignocellulose, and there has been less research done on this than even xylose. So we thought, can we do the exact same thing we did in the case of xylose and see better growth? So we basically have the same thing. We have an arabinose Regulon. we decide, in order to create an arabinose regulon, we have to engineer Gal3P to sense arabinose and then emulate all the signal-based transactions that's there in the case of galactose regulon, and finally place arabinose metabolic genes that are the galactose regulon that's there. So in our case, we already have studied this, GAL-based signal transaction. We know what needs to be done. So we basically don't have to do that. We can skip that part. So now we have only like two things we need to do. And without going into, how we did, because it was exactly the same things that we did with the case of xylose. We did the same procedure. We did a lot of different kinds of mutations here. We got a mutant Gal3P mutant that binds to arabinose better than the wild type. We thought it was good enough. We can go ahead with, engineering them. Then we placed all the necessary genes that are needed for arabinose growth in this Regulon strain, which we call ARAREG here. And to compare it, we also placed, constitutive expression strain, which we call as ARACONS. Then we compared their growth. And we see the same pattern here that, in the case of Regulon strain, we have much better growth than the constitutive strain. So this is the point, I think, when I started, we didn't want to do any more RNA sequences because we wanted more data and do more engineering on the Regulon strain before going into the, before deep diving essentially into the RNA seq of this. But one thing that kept bothering me with this is that there was this extensive need to, engineer this Gal3P sensor protein. Every time we had to engineer, we had to do like 3 or 4 rounds of engineering, which took a few months essentially, which you can think that, let's say, you want to do a catabolic engineering on new sugar. You don't want to go into this Gal3P and start mutating it every time you want to engineer one. So we thought, can we change it a little bit so that there is no need for this Gal3P engineering every time we want to engineer a new sugar for yeast. So we started deep diving into how this Gal3P works. So Gal3P works basically by 2 different regulation mechanism. One is this carbon catabolite repression that is done by glucose, and also you have activation by the xylose, galactose, or arabinose, depending on what version of Gal3 you have. So there's basically, because of all the limitations we have in mutating them, we thought, can we skip over all this and do something better? So we thought, let's create a mutant of Gal3 that doesn't need a sugar to activate it. Or in other words, it will be like always turned on. It will be a constitutive Gal3 version. And the good thing is that even though it's self-activating or it's constitutive all the time, that it gets turned on even in the absence of sugar. We have glucose, which can repress expression of this Gal3 and place it under strict tight control whenever we don't want this to be turned on. So this was not so hard to do because we can easily find constitutive mutants whenever we start doing mutations. So we were able to quickly characterize a couple of mutants, which we call here as Gal3 5.1. And basically we see here that even whatever the sugar is, you can see an upregulation of fluorescence when compared to the wild type strain that we have. And interestingly, you see that in the case of galactose, it's almost the same, but in the case of other sugars, it differs. And I'm not going over it in detail here because, I think I'm almost out of time. But yeah, you do see a similar phenomena here where there's increased growth of both xylose and arabinose with the self-activating mutants that we created. So I think that's all I prepared for this talk. So this particular section, basically we went on and did like 2 different extensions of this Regulon engineering approach. And interestingly, and even surprisingly, we found that both of them were successful. And we also kind of, shrunk this Regulon engineering, which is a 3-step approach, into like 1 step, so that it can be like easy to do for whoever who wants to do catabolic engineering yeast in the future. So with that, I think, I can just quickly go over what the thing that we need to go over is. So we were the first ones to introduce this new technique called Regulon engineering for carbon or catabolic engineering, mostly for carbon sugars. We showed that regulons are needed for growth, and using this regulon engineering approach, we can increase growth rate in any non-native carbon source that we want. With that, I would like to take any questions, and thank you all for your patience and listening to me.
Yes, so thank you, sir. Thank you for an informative presentation on Regulon engineering and how Regulons play a major role in the growth. We have a few questions here, sir. The first one is, if the HMP pathway is blocked by the Regulons, by knocking out the regulator gene, is it still possible to express the transcription factors related to the regulatory enzyme of that pathway?
But you can always, do constitutive overexpression or other kinds of things to get over the native regulation that's there. The one thing that's good with synthetic biology and model organisms like E. coli and Saccharomyces cerevisiae is that we have a host of well-paneled constitutive promoters that we can use. So you can quickly take one of those, couple it to a transcription factor that you want to be overruled by whatever native regulation that's there, and do it. I don't think it is a huge issue. Yeah, I think we can still get over that.
Okay, sir. And the next question is knockout the only process to confirm the role or significance of any Regulon gene? If there is any, can you comment on them?
Knockout is one clean way to do it, right? Because when you knock it out, you don't have to worry, is there anything remaining, anything. Another way to do it would be to, grow these in a panel of different sugars. That the idea would be that regulons are core genes that are needed and is controlled by different kinds of regulons. So when you grow them in a panel of sugars, you would see some kind of a core gene that is still being upregulated in all of them. But then you would have to control it with some kind of a basal level for that particular cell, right, or yeast strain, which doesn't have growth or metabolism happening in that. We've actually thought a lot about this particular question, and I don't think we still have any good solution to that. But I think, knockout is probably like the cleanest way to do it and the easiest. Yeah.
Okay, so the next question is positive or negative feedback loop, which gives better results?
They both have very different functions, right? Like a negative feedback loop is something that you're making more of a product, let's say, and it's might be toxic. So you signal the cell saying, stop making this right now because you have more of this being formed. And how this usually happens is that the product is what acts as an inhibitor for that particular transcription or the process itself. So that's what a negative feedback loop is, right? But a positive feedback loop is completely opposite, where let's say you are consuming a sugar, then once you detect that there's this particular sugar in the environment, then you want to consume more and more of it. You want to consume that as fast as possible. So what you do is you start cranking up all the transcription and regulation that's there, so that a lot of these more of these enzymes are being formed, so that you have increased flux through your metabolic pathway that's needed for growth or for consumption of that particular sugar. So you have a feedback loop that is positive in nature. The more you have that particular sugar, the more regulation or more positive feedback that happens here. That's what a positive feedback loop is. So I'm sorry I didn't explain that further. So they're kind of different functions. One is used to downplay something, one is used to upregulate something. Yeah.
Yeah, okay, so the next question is, will the system affect only in yeast, or is there any other better system?
Well, there are no the ideal best system that's there, right? Whatever organism you work with, you basically want to study what is the inherent regulation that's there. And then whenever you want to engineer something, we typically tend to inspire or get inspired by nature to do all of our engineering, right? Like whenever someone wanted to build an aeroplane, they thought, oh, look at these birds, how they are flying. Let's try to engineer something like this. Um, but interestingly, in the field of biological engineering, where we expect most of us to be inspired more by biology, that hasn't happened yet. So this is our, our try at, making that happen. I think that's all. Apart from that, I don't think there's any specific way to do it, or this is not like the best way to do it. In every strain, every organism that we use, I think we have to study the inherent engineering that's the inherent, regulation that's there, and try to engineer it based on that.
Okay, sir. Thank you, sir. The next question is, will dual feedback loop in Regulons suppress cellular functions?
No, that is something that's there natively, as I said, right? Like that is how galactose is metabolized. It's something that's normally there. Interestingly, dual feedback loop is something that's found in nature a lot. Because let's say whenever there is a sugar in the environment, right? You want to make sure as soon as there's even a little bit of the sugar, the cell needs to, completely, metabolize it as soon as possible. But in the case that there's a lot of the sugar, then you need to, increase your response as to a required level. So that is why there are two aspects to it. There is, the need for fast expression of these genes, but also in case there is a lot of this, there is a need for sustained and increased expression. That is why you have these two different feedback loops that kind of synergize with each other and work together to do that, to make that happen. It's actually found. I can't think on the top of my head what is used for, but it's this kind of dual feedback loop is found in our cells a lot. I think mitosis and things like that are also regulated by these dual feedback loops. So it's quite common, and I don't think it has any cellular suppression function.
Yes, sir. Thank you, sir. The next question is, was there any catabolite repression mechanism?
Yes, definitely. So whenever you're working with microbial organisms, there is definitely catabolite repression, especially carbon catabolite repression, or CCR by glucose, is something that's found. Like there's no way to get over it. We tried to, there was a different project that we were working with where we were trying to, remove this carbon catabolite repression by glucose. It's so hard to do that. Like there are like interconnecting levels of regulation that's there between them. Like there are at least like three different independent regulatory mechanisms for glucose based control. You can't get over that. You just have to work with it, or maybe we'll find a way, once you have this complete system mapped out, and start tinkering with, where you need to hit it to remove it.
Yes, sir. The next question is, can regulons from organisms which can metabolize xylose be engineered into organisms that cannot metabolize?
Yes. So that is something that we have thought of. It's a very good question, actually. But the issue is that whenever you take a regulon from a different organism, right? So a regulon basically has several functions, several ways in which it operates, right? One is the sensor has to detect this sugar, which you can easily move it to another organism. Yes, that is probably what the person who asked the question is talking about. But then there's a second aspect to it. This particular regulon has to upregulate a transcription factor, which we can also move into this heterologous organism that needs to metabolize xylose. But then the transcription factor works by binding to some kind of, binding sequence that is there upstream of genes that are needed to be, controlled. This binding sequence would be unique for that particular organism. You might have some kind of a conservation, but it is quite unlikely that you would find, hundreds of genes having the same binding sequence in front of them especially for a sugar that it hasn't even, found in its natural habitat. So I think that is the problem there, where you can get like regulation you can have transcriptional activation in the presence of xylose, but it would not translate to transcriptional activation in all the genes that are needed for growth in xylose.
Yes, sir. Thank you, sir. The next question is, can metabolic xylose be tuned through Regulon?
Who asked the question, can they explain what metabolic xylose is? I don't know what metabolic xylose, what they refer to that as. I'm guessing they're going to type. Yeah, I'm sorry. I don't know what metabolic xylose is. I don't know. I can't understand that question. Like.
Okay, so we'll answer the next. We'll go to the next question till they type the question properly. What are the computational biology softwares are available to know the metabolic pathway of yeast, mainly in Saccharomyces cerevisiae?
Computational biology. I don't think there are any computational biology softwares. You just go read a lot of papers, and I think review papers are a good starting points, right? I think review papers, there are a lot of review papers on galactose, glucose regulation in yeast, or even any particular organism, right? That's a good starting point, and then you start piecing together by reading other literature sources, where they go one particular regulation or one aspect of that regulation. You start piecing them together and then make a big overview out of that. I'm sorry, I think it would be really great if we had a computational tool for that. That's a good idea. Maybe whoever asked that question can build one if they have the expertise. I would be all into that.
Yes, I think we're done with the questions. The other person didn't type yet, and we're running out of time. So thank you, sir.
Okay.
Yeah. I'd now like to call upon our professor and advisor for this conference, Dr. Renuka Vishwanathan, to deliver the vote of thanks.
Yeah, very warm evening to all attending this conference. It's a privilege to propose a vote of thanks on this occasion, a virtual international conference on global research trends in biotechnology. First and foremost, I wish to express my gratitude to our management, St. Joseph College of Engineering, St. Joseph Group of Institutions, our chairman, Dr. B. Gopinarayanan, managing director, Ms. Supriya, director, Mr. Sachin Shekhar, for their unstinted support and encouragement to conduct this online conference. I also wish to thank our principal, Dr. B. Rao, and Dean, Dr. M. Patatavardhani, for their inspiration. I wish to thank our department HOD, Dr. G. Kumar, for his moral support, guidance, and instruction. I wish to express my thanks to the organizers, Dr. M. Chamundeshwari and Ms. Preeti, for their efforts and planning in conducting this conference. I wish to thank the speaker of the afternoon session, Dr. Venkatesh, for giving an informative presentation on engineering semi-synthetic regulons. I wish to express my thanks to other speakers, Dr. Mustafa Mohammed Issa, for his thoughtful presentation on food for thought, Dr. Jovian George for sharing his research presentation on enhancing the potency of viral vaccines, and Dr. Subuna Perumal for her informative speech on nano-composite delivery systems for cancer therapy. And also thanks to Dr. Narendran Shekhar for sharing his research experience and findings related to the current pandemic, and Mr. Sampath Kumar for his presentation on around hydrocarbon research. I wish to convey my heartfelt thanks to the team of students for their dedicated execution. Lastly, I wish to thank all participants from various places for their active participation. Thank you. Thank you, one and all.
Thank you, ma'am. And once again, thank you, sir, for taking your time. We'll now wind up with the session. I once again thank you all for attending this conference. Hope to see you all in future events conducted by our department. Thank you and stay safe, all of you. Thank you so much again.